Okay, I'd like to continue our discussion of evolutionary theory, beginning to look at the specific forces behind, the driving forces behind the genetic change in populations, which defines what evolution is. So, again, as we look at the Darwinian view of evolution, and really the modern view of evolution, the idea of descent with modification, that Darwinian idea is sound, so that we have this genetic material which goes from generation to generation, which is modified in various ways by various forces. So again, what's really important about that overall concept is the concept of inheritance, that genetic material is passed from generation to generation. So let's go ahead and look at that. If we went back, a couple of hundred years, people felt that life could spontaneously generate from non-living materials. For example, it was thought that maggots spontaneously generated from decaying meat. And of course, we now know that that's just part of the life cycle of the flies that were laying eggs on that meat. So the idea of spontaneous generation, that life spontaneously generates or routinely spontaneous generates, there's just absolutely no evidence for that. What we do know, however, is that life begets life. And again, we've talked about that. We've talked about Mendelian inheritance. We've talked about reproduction, asexual and sexual reproduction. So organisms beget organisms. Okay, that's the real nature of how organisms and how this genetic material is passed from generation to generation. So again, there's no evidence for the routine spontaneous generation of life. There's another reality here as well then, because genetic material is passed from generation to generation, and as we've talked about, again, throughout our discussion and evaluation of Mendelian genetics, the reality is that individuals that share genes have a common ancestry. And again, there's no other explanation for that. There's no biological explanation for shared genes other than a common ancestry. And again, this is something that we have talked about previously. So again, as we look at the genetic material, first of all, genetic material is universal. All organisms use DNA and RNA as the genetic material. We also use exactly the same genetic code that also suggests a common ancestry. And again, when we're looking at mRNA, 5' AUG codes for methionine in humans. It codes for that in bacteria. It codes for that in a banana. So all of this then suggests a common ancestry because we share the same genetic material, we share the same genetic code, and we share some genes. So again, we did a lot of examples of this, but if we're looking at a pedigree and we're following genes from one generation to another, you're comfortable with that. But what becomes uncomfortable for people then is what happens when that goes across a species boundary. What if we're comparing humans and chimpanzees? If we share genes, again, the only biological explanation for that is that we have a common ancestor. It's just a greater distance, a greater time period in sharing genes. And we'll talk about how some mechanisms that would explain that speciation event later. But the reality is that shared genes mean common ancestry, just as they do in our pedigrees within our own families and within our own species. Those pedigrees extend beyond species boundaries. And that's the reality of shared genes, is a common ancestry. And again, we did this over and over again. So the logic of saying, oh, it only works in this situation but not other situations is a bankrupt logic. Just as when we initially talked about if I took a distance of 10 feet, I could have done that by walking, but you couldn't do that for 100 feet, or you couldn't do that for 1,000 feet, or you couldn't do that for 1,000 yards. That doesn't hold up. Give me enough time, and I can keep taking step after step after step, and I can move a long distance. And again here, if we have generational steps, they can extend for a long period of time, and that again suggests common ancestry. So shared genes means common ancestry. And again, we know of no other biological explanation to explain that. All right, so an example then. If we look at the human and chimpanzee genomes. Recently, the chimpanzee genome was completed. It was sequenced. So again, we can compare the chimpanzee sequence of genetic material to the human sequence. And if you look at the overall difference in the two genomes, there's a total difference of about 4%. But that includes major differences such as sections of DNA that are insertions. There are some parts that are deletions between the two species. It would include individual base substitutions, and again, other differences as well. When we look at the areas of the genome that we share, however, the other 96%, there's only a difference there of about 1.2%. So when we look at our proteins, for example, almost 30% of our proteins are identical between chimpanzees and humans. And those genes then are identical. And 70% of the proteins differ by fewer than 2 amino acids. Here it says per 100, but that actually is per protein. And actually some proteins are well over 100 amino acids, so it's even less than that. So again, tremendous homology in our proteins and tremendous homology in our genetics, quite honestly. So again, that has some context then when you actually look within our own species. There's only about a 1% variation within our own species. And so we're looking at, in the shared genome, just a slightly greater difference than that. So really, it's an extraordinary similarity between the genomes of humans and the genomes of chimpanzees. And again, there's only one possible explanation for that, and it is a common ancestry. As uncomfortable as that may be for people, that is the reality. Now, we can also, again, and this is the great thing about technology, we not only can compare genes and see that the genes we have are the genes they have, but in this case, there's actually even a chromosomal comparison that we can do. And again, this idea that sometimes people will propose that, well, evolution is not testable. Well, certainly the forces of evolution are testable, and this would be an example of that. So when you look at the chromosomal makeup of chimpanzees and humans, chimpanzees actually have 24 pairs of chromosomes. Humans have 23 pairs of chromosomes. And so there is a chromosome number 2 in chimpanzees then that corresponds pretty well, or at least we hypothesized that it corresponded well to our human chromosome number 2. And so the idea was, or the theory initially was, that our chromosome number 2 was caused by a fusion of 2 of the chimpanzee chromosomes. Well, if that's true, if there is a fusion of the chromosomes, then what we should be able to see then is that there are specific sequences we associate with the telomeres, or the ends of the chromosome. So we should be able to find telomeres not only at the end of our chromosome number 2, but also telomeric sequences in the middle of the chromosomes. We should also then find sequences that we normally associate with the locations where the centromere attaches. We should also find these centromeric regions not only in an active centromere, but in some other part of the chromosome as well. In other words, we should find duplication of this if we have seen a fusion of the chromosomes. And we should actually be able to find the same genes on our chromosome as we find in the chimpanzee chromosomes. So again, when we look at our human chromosome number 2 then, we find this to be true. So again, we have the hypothesis, and then we test the hypothesis. So we think that our chromosome number 2 is a fusion of 2 of chimpanzees' chromosomes, and again, that's the reality. We find telomeres not only at the end of the chromosome, but we find telomeric sequences in the middle. We not only find active centromeric regions, but another centromeric region as well. Also, the same genes that we find on, in this case, number 2q, we find that also in our number 2, and the same sequences in the 2p, we find that in our number 2 as well. So again, there's a complete homology between those. And the fact that we have chromosomes fusing or not fusing is really not as important as the fact that the genes then are shared, which again, not only implies but demands that there's a common ancestry for our species, especially to such an extraordinary degree. It's amazing. So again, as we look at the relationship then, a lot of times people say, well, you're saying I evolved from a monkey, and that's not really what I'm saying. What I'm saying is that modern apes and modern humans have a recent common ancestor. And chimpanzees and humans have a more recent common ancestor than either has to any other living organism. So chimpanzees are more closely related to us than they are to even gorillas, orangutans, or other apes. We're their closest relative, they're our closest relative. So again, it's the same idea. Just as we follow a pedigree within a family, we can keep going back within our species, we can keep going back across species boundaries. Shared genes means common ancestry. And if you have another mechanism to explain it, bring it on, because that's what we love in science, is new things. But again, there's no known biological explanation for common genes other than shared ancestry. Okay, now, another force, important force then, is as genetic material is passed from generation to generation, random genetic change can occur. So another concept I want to address is, again, sometimes people say, well, gee, I'm not comfortable thinking that I'm some kind of an accident of nature or so on. Well, I don't know, I wouldn't frame it that way, but there certainly are forces that are random, but there are some forces of evolution that are non-random. So as we look at inheritance, that's completely random. Again, because we know from studying Mendelian genetics that inheritance follows the random laws of probability. So we know that that's a random process. Also, as we look at random genetic change, that's also something that we talked about. We can't control what mutations we generate. That is a random process. So again, the importance of random genetic change is that random genetic change generates new traits in a population. So this is where the new genes come from, new characteristics come from. And again, we've talked about this. So if you have a DNA nucleotide sequence, a change in that nucleotide sequence then potentially could create a change in the amino acid sequence at the end of transcription or translation, which potentially could change protein shape, which potentially could change protein function. And then of course, all of these mechanisms are related, but the importance there is that nature will decide the value then of this new protein function. And that's where natural selection comes in, and we'll get to natural selection a little bit later. So again, when we look at random genetic change, we've talked about all of these mechanisms before. There are point mutations, such as missense mutations, silent mutations, frame shift mutations, nonsense mutations, transpositions, polymerase misreads. And again, if you're not familiar with those, you could go back to those modules and review that. Also, gross changes in the genetic material. So these point mutations would create small changes, but a series of small changes over time can create significant and gross changes if you have a long enough time period. However, even in a short period of time, you can have massive changes due to chromosomal abnormalities. And again, we've talked about some of these. We can have translocations during meiosis, inversions, non-disjunction events. And these non-disjunction events also then can lead to duplications of chromosomal sequence or deletions of chromosomal sequences. So again, there's a lot of ways that we can scramble up our genome as we go from generation to generation. And these are random changes, and this is what creates new traits in nature. And then ultimately nature decides the value of these new traits. So again, if we look then at these processes, then inheritance is the passing of genetic material from generation to generation, and then mutation alters that to create new characteristics. And again, the value of that then is going to be decided by nature. And that's what natural selection is, nature selecting for specific genes, leading to differential reproductive success. So what I would like you to know about natural selection, though, is that it is not a random process. So nature is not random. Nature is selecting for specific characteristics in specific habitats. So it is not a random process. So again, going back to our idea we just talked about then, so changes in DNA lead to changes in the amino acid sequence, changes in protein shape, changes in protein function, and then natural selection acts on that and decides the value of that. If it increases your ability to survive and reproduce, then we say nature is selecting for that trait. If it decreases your opportunity or ability to survive to reproductive age and reproduce, then we would say that nature is selecting against you or selecting against your trait. And if you don't get to reproductive age, if you don't reproduce, then those genes disappear from the genome of your species. Okay? If you do get to pass them on, they stay in the gene pool. So again, the hallmark of natural selection then, and what defines what it is differential reproductive success. And then that leads to genetic change in a population over time. Just as new traits will lead to genetic change in a population over time. So as we talked about earlier then, again, we have population with variants A and B, where B produces 2 offspring per generation, and we could have a variant A that produces 3 offspring per generation. And if we go through the generations, if A has a better selective, if nature is selecting for variant A over B, variant A's characteristics will build up in a population over time, changing the genetic makeup of that population over time. And again, similar to what we talked about earlier, again, you could have a population where we have a certain allelic frequency. Nature selects against a particular type, and that will change the allelic frequency of that population as well. So this leads then to adaptation of organisms. Organisms will become more adapted to their environment. And again, this is something that Darwin spent a lot of time talking about because it was something that he could talk about he could document and did document case after case after case where you have a specific habitat and it modifies organisms to be successful in that habitat. Or I should say, organisms that are successful in a habitat survive and reproduce, and those genes, those characteristics build up in the population. And so again, this was kind of what he observed mostly, at least in the Galapagos Islands, where he was looking at an ancestral population of finches on the mainland of South America, yet when he looked at different islands, he found specific types of finches that were modified to the environment in which they lived. So again, specific beaks, feet, and other characteristics for those that are eating cactus or seeds or insects. So again, nature selecting for specific traits modifies populations over time. Now, again, testing this. There are a lot of ways that we can test this. We can do this in lab situations. Again, we've been doing it for millennia with our domesticated animals and plants. You know, that is essentially through an artificial selection. But looking at an example that's been done with fruit flies, looking at their reproductive success. Basically, what has been done is you can take a fly population, fruit fly population, and then deny that population food until 80% of the flies die, then transfer them to a container where food is found, so you return them to food, allow them to reproduce, collect their eggs, and then repeat that with the next generation. So in doing that, the first generation will have an average span of about 20 hours. So again, under starvation conditions, 80% will die after about 20 hours. The next generation, by selecting those that were able to survive, you can extend that average lifespan in starvation situations to 23 hours. If you run that out over 60 generations, you can extend the lifespan to 160 hours. So again, is it possible to select for traits? I don't know. Let's put it to a test. We put it to a test. We find out, yes, indeed, it is possible to select for traits. Again, just as we've been doing with all the varieties of dogs that we have and cows that we have and crops that we have and so on. That's what we've been doing, you know, for millennia, quite frankly. So then the question becomes, well, can we do that in nature? Does nature select for traits as well? And so, again, there have been situations where we've actually put this to the test of nature. So, for example, looking at a natural situation where there are predators and prey. Here the predators are a type of fish. The prey are going to be a smaller fish, basically a guppy. And so in one pond, we have a fish known as a killifish that preys on juvenile guppies. And in this case, the adult guppies are not preyed upon. And so what we find in these particular ponds where the adult guppies are not being preyed upon is that they are very colorful. So they have lots of spots and they have large spots. So they're very colorful because they don't have to be worried about displaying themselves to females and getting eaten by a predator. Now, in another pond, there's something called a pike cichlid, and the pike cichlids do feed on the adult guppies. And the more flamboyantly colored they are, the easier they are to see, and so they are subject to predation. So in these particular ponds then, the adult guppies, instead of being very, very colorful, tend to be very drab, or I should say relatively drab compared to the members in this pond. So again, here nature is selecting against bright coloration because, and again, there's a dynamic here because they need to be somewhat brightly colored to attract females, but if they're too brightly colored, they're going to get eaten. So there was a third pond where there were the killifish, but there were no guppies in that particular pond. And so you may recall that the killifish do not feed on adult guppies, whereas in this pond, they are being preyed upon by the pike cichlid. So anyway, they transferred then some of these drab guppies to this particular pond to see what would happen. What would happen if we changed the selective pressure? Would nature now select for more colorful guppies since they were not going to be subject to predation by the killifish? And so if we look at what happened then, if we look at the source population in terms of the area colored by spots, and these spots would be what are the colored areas of the fish, you notice that in the pond where the adults are being preyed upon, there were relatively few spots, or excuse me, the size of the spots was relatively small, and the number of spots was relatively small. So this would be the number of spots. This would be basically the size of the spots, or the area covered by the spots. Then, after the guppies had been transferred to the pond where they were not being preyed upon, the adults were not being preyed upon, there you can see then that after several generations, the area of coloration increased dramatically, as did the number of spots. So again, what nature was selecting for then was coloration, because then now that's an advantage for attracting females, and that increases your ability to reproduce. So can nature select for trade? Absolutely. Can we put that to a test and determine that? Absolutely. So again, a lot of times you'll hear people say that evolution is not testable, it's not true science. It's absolutely true science. We can put all of these different mechanisms to the test and determine whether they are real or not, and that's the case here. Now, when we look at natural selection, we tend to see three kind of types of selection going on. So again, if we look at an example here, if we look at a series of phenotypes, and in this example we're just using some colorations of mice. One type of evolution that we see is what's known as stabilizing selection. And in stabilizing selection, what we would have is, and again we're looking at phenotypes here, let's say that in this particular case, the intermediate colors have some advantage, some selective advantage. They are more likely to survive and reproduce, and the extremes, the light colored and the dark colored, are selected against, and that's what these arrows here are trying to show us, that we're selecting against these. So if mortality is higher in the extremes and mortality is lower in the intermediate types, again, what would you expect to happen over time is that the intermediate types would increase in numbers. And so again, that's what we observe in nature. So again, that's known as stabilizing selection. Okay, let's look at another kind of selection that we tend to see, and this is what's known as directional selection. And I'm actually just going to have you focus on this upper graph here. So again, we're going to have our phenotypes of mice from light to dark. And so in this case, what if we're looking at habitats that select for one or the other extreme? So in this case, what if the darker mice have an advantage? They're more likely to survive and reproduce, and so over time what would happen is that the population would be dominated by the darker coloration. However, directional selection means one extreme or the other extreme. So what if we had a situation where the lighter mice were favored, as we do, for example, out in our desert communities? Well, in that situation, then the lighter mice would be more likely to avoid predation, survive to adulthood, and reproduce. And so over time, then, the population would reflect an increase in those particular types of mice. So when that happens, when it's one extreme or the other extreme that is favored, we refer to that as directional selection. And then the last example is what's known as disruptive selection. And disruptive selection occurs when both extremes are selected for as opposed to the intermediate type. And so again, perhaps there's a habitat where there are patches of very dark habitat and patches of light environment. And so in this case, the two extremes would actually be selected for. The intermediates would be selected against, which is what the light arrow is trying to show us here. And so again, what we end up with then is kind of a camel hump kind of distribution at the end of that. So the extremes, both extremes get selected for as opposed to one extreme or the other. Now, in addition, there is statistical evidence and computer models which also support natural selection. And so I'm going to go a little quickly through this, but one of the first of these is what's known as the Hardy-Weinberg equilibrium. These two individuals came up with this solution or equilibrium independently of one another. And what they were looking at is what's the statistical likelihood that we see natural selection at work. So in this situation, if we let P represent the frequency of A alleles, or the percentage of dominant alleles, and let Q represent the frequency of recessive alleles, what they realized that if you added the frequency of dominant alleles and recessive alleles, that would equal 100%. And algebraically, if you squared that, that would also be equal to 1. And then if you expanded that binomial, you'd get P squared plus 2PQ plus Q squared equals 1. And so they also realized, too, that would also replicate what we see in terms of frequency of homozygotes, and frequency of heterozygotes, and frequency of homozygous recessives. So again, the idea behind this is that, again, you expand the binomial, And this replicates what we see in terms of distribution of homozygous, or it should predict what we'd see in the population in terms of frequency of homozygous dominance, frequency of heterozygotes, and frequencies of homozygous recessives. So again, if everything's working randomly, if you know the frequency of the alleles, you should be able to accurately predict the frequency of homozygous dominance, heterozygotes, and homozygous recessives. And again, the rationale behind this basically is that if we look at a population that was 60% dominant alleles and 40% recessive alleles, that would mean that 60% of the sperm would carry dominant alleles, 40% of the sperm would carry recessive alleles, 60% of the eggs would have dominant alleles, 40% of the eggs would carry recessive alleles. And if they come together randomly, as they do in sexual reproduction, or at least during fertilization, then again, we should be able to predict the outcomes here, and not just the outcomes, but the frequencies of homozygous dominance, heterozygous, and homozygous recessives. So again, if it was all a completely random process, we should be able to do that. Now, in the parental generation then, again, if 60% of the generation is dominant alleles and 40% recessive alleles, again, it would give us this outcome: 30%, 6% homozygous dominant, 48% heterozygous, and 16% homozygous recessive. What's interesting about this, though, is that in the F1 generation, what we would find is that we would actually get exactly the same frequencies. That's what we would predict if it was a completely random process. So what the Hardy-Weinberg equilibrium predicts is that there will be no change in allelic frequencies from generation to generation. And again, I don't have time right now to go into the details of this, but if you understand algebra, it's pretty easy to figure out. However, when we look at nature and we try to apply the Hardy-Weinberg equilibrium, it doesn't work. So the fact that we don't see the Hardy-Weinberg equilibrium occurring in nature tells us that there is selection of traits going on. Because for the Hardy-Weinberg equilibrium to work, it requires generally small populations. All individuals must mate, and mating must be random. But when we look at nature, are those the conditions that we see? Is everyone going to survive to reproductive age? No. And generally, is mating random? And the answer is no. Certainly in our own species, but it's not random in other species either. So again, is there evidence that nature selects for traits? Absolutely. There's abundant evidence. That's it till the next time.